But right now we have Caterina Gratton, who's a professor at the University of Illinois Urbana-Champaign. Caterina's done a lot of really important work in brain connectivity and functional networks, and she's going to be talking to us about functional networks in the brain today. Thank you, Caterina.
Thanks for the intro and for inviting me to present. It's always really fun to do these summer sessions at NeuroHackademy. Been a frequent attendee. I'm sorry not to be there in person. I was just saying I miss being out there in Seattle, and it's such a beautiful time of year to visit. For my presentation today, I've sort of arranged a kind of mix of both overview of projects that I think are interesting in the domain of what I call precision fMRI, and I'll explain that in a little bit more detail. And so these come from a bunch of different research groups to give you a broader overview of what's being done in the area. And then I'll do a little bit of a deeper dive on a couple projects that we've been doing in the lab, hopefully to give you more of a flavor of what these results and these analyses actually look like. But even though I am presenting in this remote sort of hybrid format, I do want to say I really love interactive lectures, so please don't feel shy about asking questions, and hopefully you all can help me field them if I can't see them in the moment and just interrupt. There'll also be some natural pause points during the presentation where if you have something, that might be a good moment to do, and I'll try to remember to remind you guys that that's a good time. Okay, so I'm going to go ahead and get started. And so I think many of us are sort of brought to the idea of doing functional MRI or any sort of neuroimaging research because we want to have some way of connecting our brain structure and function, trying to understand what in brain structure and function underlies human cognition. And in my lab in particular, just to give you sort of a sense of where I'm coming from, we're particularly interested in this question from the point of view of trying to understand different aspects of goal-directed functions, which are often sort of broadly under the umbrella of cognitive control or top-down control or executive functions, depending on exactly how you call them. And of course, you can study these questions at many different scales and with many different levels, ranging from the molecular to the whole brain. For what I'll talk about today, I'm really going to focus on the types of scales we can measure non-invasively in humans with functional magnetic resonance imaging. And in my work, we focused a lot on the scale of large-scale systems or networks of the brain. So different regions that are connected with one another and that we think work together to carry out common functions. You all have probably already talked about this week or seen this in your own work, but just in the interest of making sure we're all on the same page, in case you don't do functional connectivity or fMRI research, I want to talk through what I mean when I say that we measure networks of the brain. And with fMRI and other techniques, often the way that we measure this is by looking at some measure of statistical association in the activity pattern between different brain regions. So, for example, if I take the signal from left and right motor cortex over time, I get two time series from them. And across many different techniques, you could look at different patterns of similarity in these time series. The simplest of which would just be to take a correlation between the two time series and see how correlated they are. And so that's what many people do. You'd see that this left and right motor cortex are very highly correlated, whereas a region that's part of a different functional system, like a visual region, would show much lower correlations with those motor regions. Now, this was first observed many years ago, and I think what was really surprising about this technique is that it can work even during a resting state. So you can have someone sit very still inside a scanner, stare at a fixation cross, think about whatever they'd like, and yet the left and the right motor cortex are going to maintain this very high pattern of correlations. And in fact, the pattern of correlations you get during this spontaneous resting state really looks a lot like the pattern of evoked signals that you would get if you asked someone to do a finger tapping task. And so these correlation patterns seem to mimic the evoked functions of interconnected regions. And you can do this not just for motor regions, like I showed you on the previous slide, but you can do it for many regions spread throughout the cortex and subcortex. And we can represent them all at once in the form of a correlation matrix, where any given cell in the matrix is just the correlation between a pair of brain regions. I've ordered them randomly here, so this looks like noise or snow. It's hard to see much of a pattern. But actually, if you reorder the regions in this matrix in the right way, then you actually see this really beautiful structure emerge, which is called the block diagonal structure. I don't know if you can see my cursor over there. Is that okay? Yeah. Okay, great. So you can see that there are high correlations along the diagonal here and low correlations in the off diagonals. And so this is nice. It means that there's a modular structure to this network organization. And so we can use a number of different algorithms to then try to identify what these modules or networks are from the brain in a data-driven fashion. In our case, we use a technique called InfoMap, which I'm happy to talk a little bit more in detail about if anyone is interested later. But here I'm just marking off the networks that were identified with red lines. The names are on the left side that we often use for these networks. And I can also paint the brain with the different colors of the networks here shown on the left. And so in the talk today, you'll see me using both representations, which is why I wanted to introduce them. If you've heard me give a talk today before, you'll have heard me sort of say this, but I do often like to pause at this point because I think it's a really pretty neat thing that we can do here. We're using a non-invasive technique. We're using data-driven algorithms based on spontaneous brain activity. We haven't even really given anyone a task. And yet we can recover this like really detailed structure to the human brain that both recapitulates some of what we knew about brain organization, presence of visual and motor related systems that have been well studied, but also allows us at the same time to identify a lot of higher level systems that might be harder to identify with traditional techniques, systems like the default mode system, systems involved in cognitive control like the frontoparietal and singular opercular system. And so this is cool, but you might say that's super unconstrained. How well can we do this really? And I think the answer is that in group averages, we really are coming to quite a bit of consensus on the typical systems that people observe. And so you can see this in these two studies that have been very influential in the field. On the left, one conducted by Jonathan Power at WashU on 120 people. And on the right, another one collected by Thomas Yeo and Randy Buckner's lab at Harvard on about 1,000 individuals. And even though these were collected on different scanners and different people, and actually they use very different clustering algorithms, I think if you go back and forth, you'll see that they look very similar to one another. You get a very similar representation of the typical networks that we identify in the human brain on average. And so since these have been published about now 15 years ago, I think there's been a lot of excitement about our ability to use these techniques to answer many different types of questions in neuroscience. These include questions such as how do our brain systems respond when we have different forms of damage to them or we have different disease processes? How might they differ across people and do those link to behavioral traits that people have? How do they change over the course of the lifespan with developmental or aging related processes? And how are they connected to different aspects of cognition and to our changing cognitive demands from moment to moment? And I think all these questions are super exciting. And in fact, in my work, we've touched on each of them in turn and in sort of different projects. But I think that they really require that we move beyond our group average representations of these networks that I've been showing you on the previous slide to try to understand networks at an individual level. So we can understand what happens within one particular person's brain damage or disease process or their individual brain and how it links to their traits or their developmental processes and so forth. And so while this seems very exciting from the initial work, there are a number of sort of hiccups that have, or maybe bigger than hiccups, problems that have come up as people have tried to make this transition of trying to link these large-scale networks to these individual processes. And the biggest of which is that resting state functional connectivity, as it's been typically collected and analyzed, really has fairly low test-retest reliability within an individual. So this is first nicely shown, or maybe not first, but very nicely shown in sort of a very extensive fashion by Stephanie Noble and colleagues, pointing to the low ICCs and low test-retest reliability in many common studies. More recently, you may have seen this paper from Scott Merrick and Brendan Turbo-Clemens in Nature that talks about how we might need thousands of individuals to identify reliable brain behavior correlations across individuals. And lest you think this is just a problem from resting state fMRI, there actually have been very similar concerns from task-based fMRI that also seem to show very low test-retest reliability, leading some in the field to even question the entire enterprise in and of itself. Can we even do anything that would be clinically or theoretically interesting with fMRI? So I think that the answer is to change our study designs in different ways. And in fact, I think this is how the field has shifted since some of these concerns have been raised. And there are kind of two common ways that people have tried to address this problem. I imagine if that Russ presented earlier this week, he may have already talked about one of these in more detail. So I'm going to talk about the other one, hopefully to complement the idea. But the first might be to increase our sample sizes. And so if we need thousands of individuals to identify sort of robust brain behavior correlations, we should do consortia studies where we can get thousands of individuals and identify these ideas, these relationships more clearly, even when they're small. And this will be really useful for understanding population tendencies in our data. That's absolutely true. However, we and others have suggested that there's another approach that can also be useful for improving the utility of fMRI research in terms of telling us about the brain. And that's through approaches that try to increase the signal of our studies and reduce the noise, of which there are sort of many different flavors of how to do this, but many of them really rely on trying to much more deeply characterize individual brain organization and brain function. And this has, of course, a long history in fMRI. So a lot of the earliest studies in the field on retinotopy, and here I'm showing you from Nancy Canwisher in the identification of the fusiform face area, were conducted on like handfuls of subjects, I think 12 in this original study, and yet have been proven to be actually very reproducible across many studies since. And so one of the things we might consider is what was different about these studies relative to some of the other studies that we're trying to do today where we have such low test-retest reliability. And I think the answer really lies in paying a lot of attention to individual organization and trying to maximize the signal and reduce the noise within these individuals. And so on the extreme, this is sometimes called precision fMRI, especially in sort of the network field where it's only recently become popular. It's been more popular, of course, in sort of vision, sensory and motor domains for a long time. But this is also, you may have seen this under the name of deep or dense imaging, high repeated imaging. There's lots of different sort of terms that this goes by. And just to sort of emphasize how these approaches might differ from one another, I really like this graph that was made by Thomas Nasolaris for review, where a bunch of different popular data sets and largely open data sets, these might be useful for you next week as you're thinking through your NeuroHackademy projects. But a bunch of these data sets have been plotted based on two dimensions. So the x-axis here is the hours of fMRI data per participant, and the y-axis is the number of participants in a given study. And so a lot of traditional fMRI studies collect maybe like an hour's worth of data from 20 to 30 participants, of which I will say maybe 5 to 10 minutes of which are resting state fMRI. So that's the bulk of the history in resting state fMRI world is based on these maybe 8 minutes of data collection. With that first path of trying to increase our sample sizes, there's a lot of consortia studies that have really pushed along this y-axis of increasingly large number of participants in the studies, starting perhaps with the Human Connectome Project, which has 1,200 individuals, and then more recently with ABCD and UK Biobank and other groups, which are now pushing tens of thousands of individuals through their studies. But with the sort of precision or deep fMRI approach, we go down the other axis. So rather than increasing the number of participants in our study, we increase the amount and the types of data that are collected from each of these participants in order to get a better representation of their particular brain function. And I'll also just emphasize that while collecting more data is important, I think that there are also other elements to the study design that are really important to consider. This was nicely written about by Aline Coopers from Kendrick Kay's lab about some of the principles that you might want to consider in these precision or intensive human neuroimaging designs beyond just scanning more, which is maybe important in terms of maximizing reliability. But also, of course, you want to design your study well in terms of getting good tasks and other types of contrasts that are relevant to your questions of interest, optimizing the quality of your data in different ways. And so for resting state, some of the things that tend to come up, and again, happy to talk more about this in the question and answer session, but really thinking carefully about the scan parameters that you're going to be using in your data collection, thinking about strategies for improving alignments within a subject, across subjects, denoising your data to get rid of artifacts, and also how you actually are going to go about defining networks. So all of these elements, I think, are very important. And if you're interested, a while ago, I put together a tutorial for NeuroHackademy that's on my GitHub, and you can look it up in terms of different ways of looking at your data that start to get at some of these questions. And I can drop that into the Slack later on. And then the last point I wanted to make here is that in addition to how you collect the data and analyze the data, I do think with precision fMRI is maybe even more important than in other domains to find ways of sharing our data. A lot of times the data that we collect in these studies is based on very small samples. And so the more that we can collect multiple of these data sets and group them together to increase our sample size, the better we'll know that our findings can generalize and sort of be useful to the broader group. So that's sort of my introduction to precision fMRI. And then across the next portions of the talk, I do want to sort of try to convince you of why it might be useful to use precision fMRI, both in terms of improving measurement and sort of giving you some basics there, and then how that helps us to discover new things about brain function and organization, some basic properties of them and how they may link to behavior. And then I'll sort of touch on how we can even use these methods to characterize individual differences. But maybe this is my first of my pause points since I'm at the outline slide. So I'll stop here just in case there are already questions.
Any questions?
It's still pretty early on, so I'm happy to keep going. But when you see this slide, this is your reminder. Please feel free to raise your hand. Okay, so I'm going to go ahead and talk about why we would want to collect precision fMRI data in the first place in terms of measurement. And so there are two data sets that have been really influential in terms of pushing this idea of precision fMRI within the network community. The first was collected by Russ on himself. So he was scanned twice a week over the course of a year. With resting state fMRI sessions, there's also some task data as well as survey measures, blood draws, and other measures associated with this data set. And so it's one person, but very extensive amount of data. It's called the My Connectome Project, and it's available on Open Neuro for anyone who's interested. This project from Russ, which was really trying to question, can we even do this whole enterprise of resting state fMRI and get anything useful about it at the individual level? This quickly inspired several other projects, one of which I've used a lot. It's called the Midnight Scan Club Project. And so in this data set, instead of just one subject, there are 10 participants, and each of these 10 participants came in for 12 MRI sessions, 10 of which were focused on functional data. And they collected both resting state as well as a number of different task measures. And this is also available on OpenNeuro for anyone who's interested. And the tutorial I introduced earlier actually already includes some of that data too, if you want to start playing around with individual data. And so this is called the Midnight Scan Club dataset for those of you who don't know, because it was actually collected at midnight, not because they were particularly interested in what happens to your brain at midnight, but because the scanner was discounted 90%. And so, of course, we're always eager to figure out how can we fit our really high data projects into the budgets that we have. And that was the strategy used for this study. But we can circle back to sort of questions about what's happening to your brain at midnight at some point too, if people are interested. But yeah, so these two data sets have been really useful for exploring questions about the reliability of resting state fMRI and what we can do with it. And so one of the things that was like sort of done first with this project is trying to understand the properties of the measurement of brain networks, and starting with the reliability of these network measurements. So I introduced earlier that a bunch of folks, Stephanie Noble and others, have looked at the reliability of resting state fMRI and many typical data sets and found that it's quite poor. And so one question is it poor because we have too little data to measure it well? What is the maximal reliability that we will actually get from these measures? And so Tim Lauman and Russ sort of teamed up for this project to look in detail at the reliability in the MyConnectome data set. And so what I'm showing you here are two correlation matrices taken from Russ's data set from two split halves of the data, so from independent days. And hopefully what you can see is, if you look back and forth, is that these are really similar to one another. They're not only similar in the general block diagonal structure I pointed out, but even if you look for tiny little details in the matrix, they actually look very consistent across these two matrices. And if I quantify this for you, what you'd actually see is that the similarity, the correlation between these two matrices can reach above 0.95 correlation with enough data. But to get to that point of 0.95, you have to have over 100 minutes of data from a given person. And then maybe with 30 minutes, you're already above 0.9. But with 5 to 10 minutes of data, you're actually doing much worse in terms of reliability, consistent with what the earlier papers had seen. So it seems like some of the limitation that we'd had in reliability might be a data limitation. Now, this has since been replicated in the Midnight Scan Club dataset, and we've seen this also in other datasets within the lab. The exact details of how reliable things get and how much time it takes to get there does differ based on the regions that you're looking at in the brain. So the cortex tends to be more reliable than the subcortex, for example. It differs depending on the measure. You can already get kind of a sense for this. Network assignment tends to be less reliable than the correlation matrix itself. And it may differ also a bit across people. It differs depending on the data acquisition parameters, your scan protocol, and things like that. So it may be hard to have a complete hard and fast rule, but happy to tell you about what people have discovered in terms of my knowledge, at least of this space. But I do think that this approach of collecting a large amount of data is also just a good approach for understanding your data better. So even if you don't collect super extensive amounts of data on everyone, if you do it on the first few participants, you'd be able to map reliability curves like this in your participants to understand what is the maximum reliability you're getting, how much data do you need to get there, how much is it changing with increasing amounts of data. So I'd suggest it as a general way of doing things. In addition to reliability, one of the other things we should definitely care about in our measurements is the validity of the different measures. And one of the things that we might be interested in with resting state fMRI is to identify networks that have similar common functions to one another. And so we might test the validity of these measures by seeing whether or not these networks that we measure at rest actually overlap well with task-evoked activations that we collect during an fMRI scan. And so in this case, Evan Gordon did an analysis on the validity of these networks, motor-related networks in the Midnight Scan Club dataset, where he compared at rest here with the teal and the orange boundaries, with the activations during a task where people were asked to either move their hands or move their tongue. And so you can see in this individual, the evoked activations actually look like they match on really nicely to the boundaries of these networks defined during resting state. And that was relatively consistent across the different individuals in this study. But if you try to sort of match across subjects or use the group average, it matches less well. And this is true even in these primary motor regions, which are some of the most consistent. They do show some variability across subjects, but not nearly as much as some of the others, as we'll talk about in a second. So now that we have kind of good data in terms of reliability and good data in terms of validity of the capturing task differences, we might start to look again at these individual network maps and be a little bit more convinced that when we map networks within individuals, like these first 3 subjects from the Midnight Scan Club, that we can trust the differences that we see across these individuals as being something reproducible and try to understand why we have these differences and what causes these differences and what are their characteristics and so forth. So this is something that we've done a fair amount in our own work. And I'm going to talk a little bit more about this later on, but I just want to introduce a couple things that will help you to understand why we then continue to use precision fMRI studies in the lab. And so the first is that if we look at the locations that are most different between individuals and the group average, and we stack them on top of one another. So that's what this plot is showing from the Midnight Scan Club. We find that there are certain regions of the brain that just have more individual differences than others. And these tend to occur commonly in the lateral prefrontal cortex and also near the temporal parietal junction in particular. So those seem to be quite variable across people. This is a pattern we also replicated in the Human Connectome project, which is, of course, has a much larger sample of people, so it's a nice replication in that sense, but it was also collected on a different scanner and with different temporal and spatial resolutions to the scans, and so it seems like this seems to be replicable across those differences, and so we were pretty happy to see that this might be a more general pattern. And I'll just emphasize, this is also a point I'll come back to at the end of the talk. I think there's a lot of opportunities with precision scanning to try to join together these sort of x-axis and the y-axis. So to do some projects with these smaller, densely sampled data sets where you can really get to know your individual data better and trust it because you know it has super high reliability, but then to take some of those techniques to some of these bigger data sets where you can test for the generalizability of the findings. Okay, so I've shown you people show we can map networks within individuals relatively reliably, and we can characterize these individual differences, which seem to happen more in some parts of the brain than others. And so some of the types of questions that we've been asking in the lab now are about what causes these individual differences, what are the sources of them in the first place, and what consequences do they have in terms of brain function and behavior. And another question that we ask is also, once we've mapped these networks within individuals, can we find common properties that are seen even if they differ in sort of absolute organization? So is there some common relative organizational principles that are still maintained across people despite these individual differences? And so just to give you a sense of what I mean by that, we can take this region, the medial prefrontal cortex, dorsal anterior cingulate. If we look at this region on a neurosynth meta-analysis, it's probably one of the most argued about regions of the brain. Many different people have their pet theory about what this region does, and in fact, many different types of tasks and constructs seem to activate this region. But when we look at it with a network perspective, what we end up finding is that there's a bunch of networks that kind of sit near one another in this region. But within individuals, the situation is actually really even more complicated, that even though we find kind of similar networks, they're in slightly different positions and slightly different sizes across each person. And so if we analyze fMRI data the way we typically have, where we just average the brains together, assuming each voxel or vertex is exactly the same location across people, we're for sure going to be mixing together the proverbial apples and oranges. We'll mix together signals from different networks across people. And so we want to ask, now that we can sort of identify these individual differences, can we find patterns that are actually repeated across people? Like, oh, maybe we notice there is usually this like sort of motif of like purple, black, yellow that is repeated across people, even if it's in slightly different positions. All right. And so that takes me to the project I wanted to sort of dive in more deeply. And this is, again, your reminder, if you have any questions, we're just going to have a little bit of water.
Questions?
I have a bunch of these, so we can move forward. So in this section, this is the place I'm going to talk in more detail about just one project, which I'm excited about because it's a recent project from our lab that just came out in the last year. And so for this project, we wanted to focus these precision techniques on a region of the brain that is near and dear to my heart, the lateral prefrontal cortex, maybe to many of yours, because it's a sort of common target for cognitive neuroscience studies. And part of the reason it's a common target is that we've known for a long time that the lateral prefrontal cortex is really critical for a lot of higher level behaviors, including goal-directed behaviors. When you have damage to this region, you have problems with these sort of goal-directed behaviors. But despite this like very long-standing interest, it's actually still, we don't have great maps of the lateral prefrontal cortex. We don't have a clear understanding of the organizational principles of these regions. And in fact, some prominent theories really suggest that maybe there isn't that much organization to this region, that it's sort of flexibly used depending on the task demands at hand. When we look at this with sort of group average networks, what we tend to find is, yes, maybe there's like a little more organization. There's a bunch of networks here, but it's really dominated by this large frontal parietal network that you might have heard discussed. Or some models, both from the network and the task perspective, emphasize maybe there is organization, but it's really just these slowly changing gradients of organization, where you might, for example, have this rostral to caudal gradient of like abstraction of control, or dorsal to ventral gradients in terms of the content of the items of control. But the issue with a lot of these prior studies is that they use group average representations of the brain to make these conclusions. And so, as I've already shown you, this region of the brain is one of the most variable across people. And so when we average data together across different people, we're again averaging potentially different types of measures. And so this, of course, overlaps then with these locations that are commonly activated in executive function tasks and that we think of as part of these cognitive control networks. And so we really thought this was an opportunity to use these precision designs to see if there was more selectivity or more sort of common organizational principles if we address these individual differences first. And so for this project, we collected a new data set called the PAN data set, the Precision Targeting of Association Networks, also available on Open Neuro for anyone who's interested. This was really spearheaded by my former graduate student, Zach Ladwig, also a former NeuroHackademy attendee. Zach collected data from 10 participants, and in each of these participants, we had them come in for 7 to 10 functional MRI sessions. And here, what's maybe a little bit different from the prior two data sets I introduced is that we spent a lot of time trying to collect tasks that we thought would activate the prefrontal cortex based on past literature. And so in addition to a fair amount of resting state, we collected tasks across a broad range of executive function domains, working memory, selective attention, interference style tasks, both visual and auditory, and verbal and nonverbal in terms of the stimuli. And then we also collected language-related tasks, some episodic memory-related tasks, and also some social cognition-related tasks. So with this very large amount of data, we then were able to map the networks within each individual participant. And not surprisingly, perhaps given what I've told you before, we think we can do this very reliably. And so I can show that to you here with the first two participants, where we collected double the amount of data just to show the effects. And what you can see here is that the networks from the two split halves of these participants are relatively similar to one another, and they look quite distinct across different individuals. And so that's true for the network measures that we get from resting state. But we show also that we can do the same thing with the task data too, in this case. And so again, hopefully you can, if you go back and forth, you can see that the task measures look quite consistent across split halves within a participant, and they look different in the fine scale features across the different participants. And so knowing that we have this reliable data, we kind of went back and tried to interrogate what does the individual prefrontal cortex even look like relative to our group average models? And so we'll start with the resting state network data. And I'm showing you here on the left a typical group average model, which, as I said before, it does include multiple networks, but has this very large yellow frontal parietal swath. And when we look at our individual participants, what we see is something a little different. So the individuals look more complicated. They have a lot of different patches or segments from many different networks that are kind of tightly packed into this area that aren't observed in the group model. And so this has two consequences if we quantify it. One consequence is that the frontal parietal network, shown here in yellow, we are overestimating it for the most part. In most individuals, the frontal parietal network is smaller. It's still a big network, but it's smaller than you would expect based on those group average representations. The other is that we're also underestimating how densely packed in networks are, and particularly within the prefrontal cortex. And so when we can count this by just taking each position in the frontal lobe and just counting how many networks are in close proximity to that location. And if we do this in the group average and in this one subject, what you see is that the group average maybe has a high density of networks along the more caudal regions. These are also the regions that don't differ quite as much across people, so maybe not surprising. But you lose a lot of that high density of networks in the more rostral regions that you still maintain in this individual participant. And this is actually very consistent across all 10 of our subjects. So you can look at the average of these density maps and see that there's this high density region in that more rostral portion of the prefrontal cortex that's being systematically missed in the group average models. And so what's happening there? When we look in more detail, what we find is that in this region, there is, in the group model, a big piece of the cingulopercular network. This has been reported for many years, back to the original definition of the cingulopercular network. But one of the things that we see if we look in other parts of the prefrontal cortex is that the cingulopercular network is often flanked by the dorsal attention and language network in kind of this conserved motif. And this is true also in other parts of the brain. But that seems to be missing in this more rostral region. But when we look within individuals, we actually find that that same motif is present and it exists again, dorsal attention, cingulopercular language. That seems to occur. And we got it in 9 out of our 10 participants. One participant was missing their language region, but if you look at the functional connectivity of the language network, you actually do see evidence that there is also functional connectivity to this region. So I think it's just being missed by our hard parcellation approach. So this seems to be quite a consistent motif that we can observe because of the individual mapping. It's a relative motif. It's not occurring in exactly the same place or in exactly the same position across people, but it is common across all of these individuals. This motif is actually embedded within a larger sort of structure that seems to be happening here in the rostral prefrontal cortex, where not only those 3 networks, but pretty much all of the higher level networks seem to come together at this cingulopercular network, where you find the cingulopercular, dorsal attention, language, but also the 2 default mode networks, the salience network and the frontal parietal network, all in close proximity to one another. So this is a discussion on networks so far, but I promised you that I think this will tell us something about brain function too. And so I wanted to turn to the responses during the task activations of all those tasks that we collected inside the scanner. And I'm going to start with 3 tasks that have been more widely studied by Rodrigo Braga and Ed Fedorenko and Lauren DeNicola and colleagues, where they have in the past shown that these tasks tend to have a pretty good match to specific networks. And in fact, they use these sort of localizers for these tasks relative to these networks. So these visual and auditory language tasks, where, for example, you compare sentences to non-words that you're reading, tend to have activations that strongly activate the language network relatively selectively. If you do theory of mind related tasks related to emotional pain or false belief stories, you tend to have activations that are relatively selective to the default network B. And if you do episodic projection tasks related to thinking about the past or the future, you tend to find activations that are relatively selective to default mode network A. And so they've shown this in the past for the whole brain. It's also true we see here in lateral prefrontal cortex specifically. And in fact, if we look in a little bit more detail at these maps, what you'll see is if you compare the patterns of these individual networks for, say, the language network compared to the group average language network, that the activations within that person are much more tightly matching onto their individual network maps than they are to the group average maps. And in some cases, the group average maps are completely missing the actual activation in that person. And so we can quantify this. There's a better overlap within individuals between their resting state networks and their task activations than there is from the group average or other people. And interestingly, this is actually particularly true in the lateral prefrontal cortex. So if we compare this at individual advantage in the lateral prefrontal cortex, kind of non-lateral prefrontal cortex regions of the brain, you see it's even larger here, probably because of the high level of variability in this region. The last point I want to make is also, even though we see common motifs, we do see unusual idiosyncratic brain organization within these individuals, and we still see this nice match between individual networks and task activations for these idiosyncratic regions. So for example, 1 in 4 of our 10 individuals, we find a piece of the language network that sort of cuts across the classic frontal parietal network shown here. So we don't see it in the others, but we see it in these 4 people. And if we look at the task activations in these 4 people, we find that that language piece defined with resting state really does show activations during the language tasks. And does not show activations during spatial working memory tasks, which really nicely skirt around this region. And so this really suggests that to understand the functions of the lateral prefrontal cortex, we need to be able to account for these common, widespread, weird, idiosyncratic patterns of network organization that we find in this region. And so just to make that more concrete, let me present to you a scenario where you're doing an analysis where you really care about the frontal parietal network and how it's responding in a task. So you put a region of interest squarely in the middle of what you think is frontal parietal network territory. And you look at it, and in a subset, a fairly large subset of your participants, you're actually going to be hitting a lot of language network in that case, not just frontal parietal network. That means you're going to be missing a whole lot of spatial working memory task activity and instead capturing a bunch of language task related activity. And so that's going to potentially lead you down very misleading roads in terms of interpreting your results. So those were the kind of common higher level tasks related to language, memory, and social cognition. But we also collected a bunch of task activations related to more executive function related processes. I mentioned these before. They're visual and auditory, working memory, interference and attention tasks, verbal and nonverbal. And when we look at the activation maps in these participants, what we find is that the activations tend to be quite patchy, interestingly. So they're not activating the entire lateral prefrontal cortex, they're activating subsets of it. There are maybe some common patterns, but they also look fairly distinct in the high level details across people, but they look quite consistent, surprisingly, actually, across these very different tasks from one another. And if we overlay these on the network maps, what we see is something a little different from before. We don't get this nice like one-to-one match in the same way that I was showing you before of strong selectivity for just one network. Rather, the activations in these executive function tasks tend to activate at least 3 networks quite commonly: the frontal parietal, the dorsal attention, and the cinguloopercular network. And instead of sort of sitting in the middle of the network, we actually find that these activations often are lying kind of at the border between these networks. And in fact, we can quantify this. They're significantly more likely to happen at the border than further away from the border, which is the opposite of the pattern that we see for the other tasks. So I think this is interesting because it suggests that maybe this gives a functional significance to why we'd want to pack in so many networks into the frontal lobe. Why do we have this patchy organization? It suggests that maybe this organization is useful for these cognitive control functions. Maybe there's some sort of information integration or handoff that's happening at these border regions. Okay, so those were a bunch of results from this one project, but just to summarize for you, showed you some evidence that when we look within individuals at their prefrontal cortex, we see differences from the group average. They have a much more patchy and complicated organization with lots of borders between networks. Despite this, we can find common motifs, and these tell us about patterns of task responses in these folks, with networks following the task responses very well for language, theory of mind, and episodic cognition. Whereas cognitive control activations seem to happen most frequently at the borders between some of these higher level networks, suggesting again a functionality to these network borders within that context. Alrighty, so that brings me to the end of that paper, but I do want to mention that these sort of techniques have also been used to tell us, discover a lot of other really important basic properties of brain networks that have been really influential in the field. And so, for example, from Rodrigo Braga and Randy Buckner's work, they use these sort of methods to show that what we commonly have called a default mode network in the past is actually probably two fairly separate networks from one another. Already heard me mention this in the earlier part of the talk, but they really don't overlap that much within individuals. A lot of the sort of overlap that people have tended to see is likely because of smearing of signal across individuals. Scott Merrick and colleagues use this approach to look in detail at the organization of the cerebellum, and where they find that the frontal parietal network tends to be larger than expected in the cerebellum relative to how big it is in the cortex. And Deanna Green has used this sort of approach to look in again at subcortical structures of the basal ganglia and thalamus, and trying to look for network loops that might be relatively more selective for particular networks or have more integrative functions connecting to multiple different networks. So these are just some of the possibilities of what you can do with this work. Another very influential paper that has come out from the precision field is also just discovering a completely new network in an area of the brain that we thought is relatively well described. So many of us think of the motor cortex as having this classic homunculus of responses for different parts of the body. And Evan Gordon has observed as he went through that this seems to not be exactly the case with the networks, where you find actually interconnected set of three different regions that respond to whole body movements that are more highly connected with higher level parts of the cortex. And this has been called the somatocognitive action network. So hopefully those projects give you some ideas of why you would want to do precision fMRI and how we can still do reliable and replicable science with smaller samples if we really try to maximize the signal that we're getting from each of our participants. And so in the next part, I want to turn to how we might connect these to behavior as well. Again, I'll take a quick pause because my throat's hurting.
Yeah, we got a question in the room. 1 second.
Yeah.
Hopefully you'll be able to hear this using the microphone, but if not, I'll repeat it for you.
Hey, really interesting so far, thanks. I was wondering, so if there's so much variability in the networks between individuals, then how do you go about like classifying them in a uniform way? Is that just like with all the task data to identify like exactly which network is in a particular location relative to like other individuals?
Yeah, that's a great question. And I think I heard it also. I don't need it repeated necessarily. It's a tricky problem. I guess there's kind of a couple levels of the way that I would answer this question. Let me go back to the original network results. So the first part of the answer to this question is that we haven't yet found a lot of super clear examples of cases where we have a completely absent network in one person or a completely new network in one person that isn't found in other people. So once you go to the network level, typically you do find a frontal parietal network in everyone. So that already helps with the matching. You're not sort of doing an end to end where you have no idea, N to K, no idea of how to match them. And they do have somewhat related organizational patterns. So you will tend to find a part of the frontal parietal network that's in sort of the lateral prefrontal cortex. And so often in practice, the way that we do this is through some sort of template matching where we find networks that have enough pieces in the right areas that they will match on to templates that we find across people. But as we get to smaller and more specific networks, I think there is also, we're going to have to improve our sort of methods for matching networks across people that incorporate some of these ideas about relative organization that I was hinting at, these sort of common motifs. And I know in talking with Rodrigo Braga already that that's something that his lab commonly does, for example, for the two default networks, which do always sit side by side and can sometimes be a little bit confusable. That there are some common patterns that you'll tend to observe. Like in this case, default network B tends to be a bit larger in general in the frontal lobe, and it tends to sit a little bit more forward, I think, relative to default mode network A pieces. So kind of once you find two of them, you know the one that's kind of sitting more rostrally is more likely to be B, the one that's sitting more caudally is more likely to be A. And there's a lot of rules like that that you can observe if you look at enough of these brains, but we need maybe more systematic ways of writing those down and characterizing them. Hopefully that kind of gives you an initial picture. It's something that we actually wrote about a little bit more in detail in a recent annual review article that I'll put at the very end, that he has like a whole section about this exact question.
So Caterina, I actually have a question. I've always been struck by these, the way that the brain will have areas that are very consistent across subjects, and then an area that consistently across subjects is only consistent for that subject, such as the lateral prefrontal cortex here. And it just strikes me that one way to explain this is that there is some kind of biologically constrained scaffolding in the brain, and then regions that depend a lot on experience. And so I'm wondering, and apologies if this is kind of tangential, but I'm wondering if you can say anything about the heritability of this region.
Yeah, I actually am going to show some slides on this later on, but I think it's a really fascinating question. It's a little hard to fully test it, but it sort of gets into this like what causes this individual variability, and there's different theories about it. I will say, so there's some evidence that these individual differences in brain organization and brain from our work that I'll show you later are heritable. In the sense that they're more similar among identical twins than they are relative to fraternal twins and relative to unrelated individuals. Others have also shown that network properties have that sort of heritability aspect to them. And so presumably some of these differences may be due to differences in underlying genetics and sort of allelic differences across people. Some of the ways that I think about what allows for and what causes these individual differences, so they're also, of course, not perfectly heritable. So there's going to be some experience dependent effects in there. And some of the ways that I think about these have been driven by some of the theories that have been put forward by Randy Buckner's group. So there's an earlier theory from him and Fenna-Krienen, which talks about the idea of how do cortical areas come about. There might be these transcription factor sources that help through gradients to define the sort of general organization or the template organization of the human brain. But as you get further from those original source locations, those transcription factors are released, the gradients get shallow. And so there's less of that sort of biological constraint on how these areas can be positioned exactly, and it allows for perhaps more experience-dependent effects in those regions, which I think is reasonable. It doesn't explain, though, exactly why we get these common motifs in these areas. And so one of the other theories I quite like is one that talks about also the idea that we might all start with these larger proto-networks that then fractionate and sort of differentiate from one another with experience. And so this is a paper from Lauren DeNicola. And so that would explain why, for example, the two default mode networks are always next to one another. Maybe they start early in development as a single larger network that then separates based on sort of differences in experience, and they sort of may actively fractionate from one another. And it also fits with some data that exists showing similar effects within visual regions of sort of proto-organization that then gets refined with time.
I see that one of our online participants has their hand raised. So Sophia, if you want to go ahead and ask your question.
Hi, yeah, super interesting. And I know that you addressed this already, but in the individuals where you're seeing that like some of those lateral prefrontal clusters aren't representing like one of the networks. Could you re-explain your interpretation of that, whether you think that those networks perhaps don't exist as clusters in those regions, or is it more so methodologically somehow they're just not being identified?
So do you mean like the language example I gave with like the idiosyncratic regions?
Let me see.
I can go to the slide and if that triggers
Probably.
this one, maybe.
Maybe. It was there was something where it was like 6 out of 7 of the networks were identified. It was more of the
Oh, this one.
this one. Yes, correct. Yeah.
Okay. Yeah, so here, I, it's a little hard with such a small sample to know whether the differences that we're seeing across people are exactly like our true individual differences or are a function of our method. So in 8 of our 10 people, we saw all 7 networks come together, and in 6 out of 7, in 2 of the people, we found 6 out of the 7 networks come together. And I think in this subject, the one that wasn't there is the language one, which I showed you does actually kind of exist. It's just not quite being captured with our hard parcellation method. So I do think that's a methodological difference. I think, honestly, it's been a little while since I've looked at this subject, so I can't remember if it was the case for this other subject as well. We did replicate this result also in the natural scenes data set. That's also not a huge sample, but again, another example of trying to use multiple of these precision fMRI data sets to put them together to get a little bit more participants. And there, I think all of the 6 participants, if I'm remembering correctly, showed all of the networks come together. So I suspect it's a relatively general principle, but I think it's going to be a really interesting question then. Do we find cases where we really are convinced this person does not have a piece of that network? And what is then the functional significance of that? Like, are there any correlates? And I don't know the answer to that yet, but I'm really interested to do this sort of work in larger samples to get at that.
Awesome. Thank you.
We got one more question in the room. 1 second.
Hello, thank you very much for your presentation. I'm kind of asking a question that's less about the individual aspect. I just wanted your opinion on our lab does a lot of resting state analysis looking at how resting state networks relate to behavior and cognition. And one thing that we struggle with this moment is the naming of each network and what kind of taxonomy is the best to relate to, I suppose, cognition and behavior. And I know this like in, I think it's Lucinda Udin's paper, are used for like a more anatomical naming. So like say the mid-singular insular as opposed to the salient network. And I just wanted your opinion on kind of how you decide a network taxonomy. Yeah, does that make sense?
Yeah, absolutely. And I think it's an important question because I think it stymies progress in the field when we're not all talking about the same thing, or we are talking about the same thing, but we're calling it different names. And it's kind of unfortunate that it is the case. It's more true for some networks than others. So I guess I was part of actually a group that Lucina then ended up running after she published that initial paper to talk about this issue of network taxonomy and sort of how do we talk about networks in a common way. And unfortunately, I don't know that we ever came to consensus on the right names to call things. I also really like anatomical naming for networks, but I do think it's challenging because they have pieces in multiple different places, and those can differ across people. And so you get to have these like 7 long names that might be kind of hard to remember. And so the functional names do tend to be sticky in that they're easy to remember and easy to say quickly, but they of course have the downside that they seem to ascribe a function that we may or may not think is truly happening uniquely in that region. Default maybe being one of the most common and most argued about over naming conventions. And so I don't know that that gives you an exact answer. I think my instinct would be to go anatomical, but with this caveat. I mean, it would be kind of nice if we could name them after like Pokemon characters or something that would also be similarly memorable and but distinct from one another, and leave the question of function to the science to tell us about it. But I did want to mention that one of the things that came out of this initiative that Lucina ran is a paper that I think Ruby Kong is the first author, where she created this toolbox that allows you to more directly compare a bunch of the large atlases in terms of you can like input your map and then you can say which network, what would like the Yo Atlas call this network, what would the Power or Gordon atlases call this network, how similar are these networks really to one another. I think the answer there is actually most of the networks are quite similar to one another, which is nice. A few of them kind of split into two, but the naming can be quite different across them. Salience and singular opercular maybe being the most argued about. I don't know. So happy to have more discussions about that.
We got another one in the room.
We're talking one of the most exciting subfields of FMRI, I think, right now. So I'm not super familiar with the literature, so it's not like a literature question. Is there any research trying to validate or compare these individual functional parcellations with, for example, an individual structural connectivity with their tractography? Yeah.
Yeah, I love that question because actually Ariel and I are collaborating on a project that is trying to do something very related to that. There are some other projects that have tried to also compare structural and functional connectivity to try to validate. And the general answer from the prior projects has been that they're correlated with one another, but not super well. Often like 0.4, 0.5 has been kind of the correlation value that I have in my head from some of the bigger studies that have done this. But most of those have been done at the group level. So, you know, part of what Ariel and I are interested in is could you find better matching at the individual level and could they be used to validate some of these smaller pieces? This has been done a little bit in pieces. So, in Russ Poldrak's and data set, he looked at some of the tractography to functional mapping there. And I think what was kind of interesting is they saw actually the more data you collected, the better the mapping between the functional connectivity and the structural connectivity, probably because it allows you to overcome some of the noise in the functional connectivity. It also allows you to do partial correlation, so to address some of those indirect connections, which of course functional connectivity usually mixes in, but it's hard to do with too little data because you have so many regions, so it's hard to partial everything out. So I think that really helps with the functional connectivity. Of course, structural connectivity also has its own issues in terms of crossing fibers and other things of that sort. So I'm not sure they will ever be a one-to-one match, but I definitely think we're going to get a lot of value from putting the two together. I guess one other thing to mention is that people have also looked a little bit not at just tractography data, but other structural features to see if they line up. And so, for example, in Evan Gordon's original paper on the Midnight Scan Club, they showed that the transition of the T1, T2 ratio, the myelin sort of ratio, really actually corresponds quite nicely to network transitions within individuals in the area close to MT. So there's, I think, reason to think that these can also be validated in other ways.
Okay, I think you can continue now.
Yes, still have more to talk about. Don't worry. Or maybe you are worried because you all have heard a lot of talks, but you will hear more. Okay, so the last section was less, or the next section is a little bit less detailed on our own work, but I wanted to talk to you a little bit about some of the cool projects that have been done looking at using precision fMRI to link these measures of the brain to behavior. And there's kind of three that I wanted to highlight. The first is looking at using this sort of approach to look at responses to brain injury. The second is looking at responses to interventions. And the last one is looking at responses to hormones in the brain. And so the first project I think is pretty cool. It's actually the one that originally inspired Nico Dosenbach to do the Midnight Scan Club project. He's a pediatric neurologist, and he had this case of a kid who was referred to him. He was playing competitive baseball, and his coach had noticed that even though he's a very good baseball player and great with his left hand, that his right hand was pretty weak. And so he got referred up to him, and eventually they scanned him. And they discovered that this is what his structural MRI image looks like. So he's missing a very large amount of his cortex, including most of his motor cortex on both sides, entirely on the left side and almost all of it on the right side. And it's pretty amazing. Despite all this, he's able to play competitive baseball, and he was doing just fine in high school, just the same as his siblings had been doing. You can see him here in a mechanic shop. That was like the job that he was taking up at the time. And so I think you hear these cases every once in a while of these like really extreme forms of plasticity that can happen with brain damage. In this case, they think likely he had a perinatal stroke. So right after he was born, he may have had a stroke that led to this damage, but his brain was able to recover from it. And so precision imaging is really like an ideally suited case for this. You don't have, 100 or 1,000 or even 2 subjects necessarily with this kind of injury. You have 1 subject. And so when you want to look at brain reorganization, in this case, you really want to look in detail at that 1 participant to see what you can learn. And so Tim and Nico did these analyses where they wanted to map the motor networks first and then networks more generally in this participant's PS1's case. And so what they saw when they did resting state network mapping, looking for the motor network boundaries within this participant, they saw that in the right hemisphere, there was a tiny bit of spared tissue, motor tissue. And this is the sort of activations that you get in that tissue. And this corresponds to a very small portion of the motor network that seems to be remaining and is yet allowing him to be a competitive left-handed baseball player. On the left side, what you find is that actually the motor activations and the motor network map are displaced. So they're now in the post-central gyrus instead of in the pre-central gyrus. And he's still able to move that side of his body, so they seem to be functional. And actually, this is another case where they did some tractography and they saw that the cortical spinal tract also seemed to have been displaced in the same direction. And so it shows that even though you may not get complete functional retainment with this change, they're still, he's still able to co-opt this other part of the brain for motor function. And so this is, I think, really exciting about our ability to use these techniques in clinical applications to understand what's happening after injury. You can also then use it not just to look at the motor-related networks, but how do all the other networks sort of reorganize in response to this injury. And sort of one of the things that they saw is that, interestingly, the networks seem to try to maintain their overall network size. Like proportionally, each network was about the same size as it is in other people, even though there's a lot less cortex to fill. And so maybe there's, again, some principles about sort of give and take across the networks that causes that organization to try to be maintained. So that was a cool case. Another graduate student from Nico's lab, Dylan Newbold, also used these sort of approaches to look at interventions, in this case, plasticity-based interventions. So some of you may know that when people have strokes, one of the common sort of interventions that people use is actually to put a cast on the arm that the person can already use to force them basically to use their injured arm more. And so this is a common intervention that's used in neurology, but it's also kind of just an interesting test case of how much can we even change functional connectivity over time. And so what Nico and Dylan were able to show is they scanned 3 people for 2 weeks before, during, and after putting on a cast. So 6 weeks total. And when they looked at the connectivity of these motor hand regions, what they saw is that before the cast was on, the connectivity is really high. This is actually one of the highest connection values you'll find generally in the brain. You see the motor cortex and you'll get the homotopic region in the other hemisphere. But this drops down dramatically during the cast period and then recovers after the cast. And so you can actually see this in like the individual scans within these participants during this period of time. So this is a pretty cool proof of concept that you can indeed change functional connectivity with different types of interventions. And so I think it sort of is a nice proof of principle that you could do this to study other types of interventions and how they change within person network properties. And another type of not intervention, but sort of natural design of looking at brain dynamics within a person using these sort of precision approaches has been really spearheaded by Emily Jacobs' lab. Laura Pritchett is sort of as the first author on these very influential papers, which looked at how brain networks might change with the influence of different hormones. And in the case of these first couple papers, different hormones over the course of the menstrual cycle in one woman. And so they were able to show connectivity patterns that correlated or related to these differences in hormone levels over the course of the month by scanning one individual every day over that course of that month, and then again one more time. So it's a pretty neat data set. It's also available on Open Neuro for those who are interested. I think they have another project that is also on there. I haven't checked. I should check it. That is on one man with morning evening scans. So also looking at testosterone differences. And then very recently, they've also had one project where they scanned one woman during the course of her whole pregnancy. So they had scans from her before pregnancy, during the pregnancy, and then postpartum. And so again, we're able to look at differences in the brain, in this case, largely on structural measures in terms of, for example, gray matter thickness and the white matter measures and things of that sort, where they again see these pretty dramatic shifts in a brain structure during the pregnancy, some of which we normalize afterwards, not all of which necessarily. And so I think these are really fascinating cases where it'd be hard to do these studies in very large samples, but by doing these really detailed measurements and getting a lot of measurements before and after, you can kind of compare within a person and how things are changing to get new insights about brain behavior relationships. I wanted to also mention that I think that a lot of folks are interested in how can we do this better and how can we do this across bigger studies. And there are a couple great efforts out there. One is from Bertrand Thirian's group called the Individual Brain Charting Data Set, which has data from 12 participants who each did 150 common task contrasts. So you can use that to look in more detail at the individual brain. And the other of which is from Kendrick Kay, who I think has been or will be a speaker at NeuroHackademy, a natural scenes data set, which contains 70 data from 8 participants who each did 40 hours of fMRI, largely focused on natural scene processing, where over 73,000 images were presented to participants. And I think these have been super influential. Natural scenes data sets being used in lots of different projects to try to improve our understanding of visual processes in the brain and also to improve AI algorithms of vision. And so I think as we have more of these data sets that we'll continue to see the influence on that. And then the last thing I want to mention in this section is that while I think we've made a lot of progress in improving our understanding of how can we get better measures of brain networks, at least to get more reliable measures, it's also important when we want to connect brain and behavioral relationships to think about the behavioral measures that we're collecting as well. And unfortunately, just like with the brain measures, when we do very large data sets, we often don't get enough data to really get a good measure of behavioral variables. And unfortunately, that's particularly true of a lot of the behavioral variables we care the most about, like inhibitory control, which is often measured with tasks like flanker task or Stroop task. But in these data sets, you might collect something like 40 to 50 trials of that task to get a measure of a particular person's inhibitory control. And so in a project spearheaded by 2 postdocs in my lab, Hae-Jin Lee and Derek Smith, we set out to use kind of similar methods to try to understand variability in inhibitory control and how precisely we can even measure it behaviorally. And what they show is that you can get trait-like measures of inhibitory control, but it does require more like 1,000 trials to collect it really reliably within a person. Of course, different people can have different tolerance for different degrees of error. But if you really want to know about that person's inhibitory control, because you say want to connect it to their ADHD or to some other clinical variable, that you might need to go about it in a way that allows you to measure it really reliably as well. And so that data is also available online for anyone who would want to play around with it. So that's, I think, like just a snapshot of what you might do with precision fMRI in terms of connecting to brain behavior relationships, especially improving within person sort of relationships. I think there's a lot of potential in that case. But the last thing I wanted to turn back to is the idea of how we use these techniques to characterize individual differences. Here, I think, is a place again where we really want to combine with that big data. Use the precision data to really make sure we have really reliable measures, and so that we feel confident about the individual differences we're measuring, but then ideally test them in a larger data set so we can look for the generalizability of these results. And so that's something that we've done in a couple of past projects. I mentioned one earlier. We used both Midnight Scan Club and Human Connectome project data to measure these individual differences. Here we just create these continuous maps of how similar each person's brain is to the group average, here for subject 2 and 6 in the Midnight Scan Club data set. And we can show that we can make these measures really reliably. Ooh, they come out super consistently across different sessions in the Midnight Scan Club dataset with these blue locations that are very distinct and dissimilar from the group. These are locations that we call network variants for the purposes of this project. One of the things that we just did very recently, I just want to highlight because it's a new result from the lab, is to also look at how consistent these are, not just over a few weeks that took us to collect the Midnight Scan Club data set, but much larger time spans. And so we were lucky enough to have data from 3 participants who had a fair amount of resting state data across very large windows, one with 8 years apart and 2 with 13-year apart data sets. And so, and then we also have 7 additional participants have 1 to 3 year gaps in our data. And I think really critically, we have a lot of resting state data, so we can overcome some of the noise issues, the test retest issues that I talked about at the very beginning of the presentation. And so, and then I forgot to mention, but Russ also very kindly shared the new data from the MyConnectome project, where he's recollected data 10 years later. So we have a 4th data that we were able to use for replication in this work. And so what we find if we map the networks in these individuals across these different time points is, I think, really a remarkable degree of consistency, even over these very long time spans. And so if you look at the brain networks from this person 13 years before to after, here we've just colored in with colors any regions that are consistent. Gray is inconsistent across the different time points. Hopefully you can see a lot of the brain is colored in. And if you zoom in actually on the prefrontal cortex, which I told you to focus on before, you actually see a lot of those like really idiosyncratic details are very consistent even over these very long time windows. And this is true for these 4 participants. It's true even though over these time windows, there were a lot of changes that happened to these participants. They had career changes, they moved, they had children, they lost parents. The pandemic happened in the middle, which was a very major social emotional intervention for all of us. And yet a lot of properties of our brain organization are kind of staying the same, suggesting to us that there must be like some homeostatic mechanisms that are really helping to maintain the structure in the face of all these changes. Now, these are all young adults, so I'll point out that some of the things we're interested in doing in the future are to look at changes in other age ranges, periods like adolescence or older age, where you might expect to be transition zones in terms of changes in life. But at least it sort of sets a benchmark for what could happen. So in addition to characterizing how stable they are over time, we've also used these to characterize other properties of these individual differences. I'm not going to talk in a huge amount of detail about them. I'm putting up a couple papers that we've published on these where we show that they, again, occur consistently. They predict these individual differences in task activations across people. But I did want to mention one recent project that was spearheaded by former research tech in my lab, Ali Duretsky, where she also asked, are all individual differences the same? And one of the things that we tend to see when we look at these individual maps is that some of the individual differences are what we might call border shifts. So this yellow network in subject 6 taking over part of the territory that would be part of the purple network in the group average. So frontal parietal network is a little bit expanded in this person. And so you might expect that these are the majority of the individual differences that we find. But actually, we also get clear examples of things that don't fit with that profile. So like islands of this like red default mode network that's sitting in the middle of the frontal parietal network, not close to where the frontal parietal network typically is. And so we call these ectopic intrusions. And Ali went through and sort of characterized border shifts and ectopic intrusions across multiple data sets, showing that actually, surprisingly, ectopic intrusions are relatively common. We have them in most people. Those might be related to those patches I was talking about earlier. They do seem to differ in their properties relative to border shifts in terms of where they're tending to occur the most and which networks they're linked with. But the one I wanted to tell you about, and we sort of alluded to earlier, is they also seem to differ a little bit in their heritability. So here we use data from the Human Connectome project, where we were able to compare network variance among the identical twins, fraternal twins, and siblings, and unrelated individuals. And if we look at them, we always see the sort of general heritability pattern. So they're the most similar among identical twins and least similar among unrelated individuals. But you see that this pattern is stronger, sort of more closely tracking with genetics for the border variants than the ectopic variants. And so to us, this suggested that these ectopic variants might be a little bit more affected by these experiential or environmental variables relative to the border variants, which might be tracking a little bit more with genetic differences across people. This has also been used in sort of clinical applications. There's a paper came out a couple of years ago now that made a big splash from Chuck Lynch's group, which used individual network mapping in depression. And what they had is actually 3 case examples where they had scanned individuals with depression many times over the course of the year. And they had sort of just observed and were surprised by that when they mapped the networks within these individuals, that the salience network seemed to be really large in these individuals relative to what they typically had seen in healthy controls. Now, this was an unexpected observation, so the first thing they did is try to replicate this in other data sets, and they found that in larger data sets, they still see the same pattern of an expanded salience network. They actually show that this seems to precede depression in the ABCD data set, so they see it at earlier ages than when kids then later show depression. And interestingly, it's connected back to the ectopic and border shift results that we had. They showed that this was largely driven by border shifts of the salience network in these individuals. So maybe a biomarker of risk for depression in these folks. In our own lab, we're interested in sort of expanding beyond some of the populations that have been used. A lot of our initial results in these precision data sets have been in very homogeneous samples, young, neurotypical adults often associate the lab or something of that sort. And we really need to go beyond that to be able to generalize our results more. One of the ways that we're interested in doing this is to go across the adult lifespan and try to understand networks, not just in young adults, but how they change as we age, where we see really different outcomes from aging. I want to highlight some of the folks working on this, and in particular, Young-In Park, who I think might be sitting there in the audience today, who's been working on these projects in my lab. So you can talk to her if you're interested in this data. And then we're also, in a related fashion, interested in what happens with neurodegeneration. And here we've worked a lot with Megan Campbell's group at WashU to look in Parkinson's disease and what happens when networks start to break down in Parkinson's disease. And I think this is a really exciting area. There's been some neat work from Hessian Blue's group showing that, for example, that new scan network that was newly discovered might be particularly connected with symptoms of motor symptoms in Parkinson's disease. And then we in our project are interested in trying to understand how these might relate to other non-motor symptoms in the disease as well. So hopefully these examples kind of give you, inspire you or give you some flavor of what you could do with these smaller precision fMRI designs. and sort of come back to this idea that we have a lot to gain, I think, in the field by thinking about how do we do our science better. Of course, being at NeuroHackademy, you guys are already thinking about that and learning about that. And I think thinking about these different techniques and ways of improving the reliability, improving the generalizability, and then ultimately the reproducibility of our science will sort of bring us to in a long way to make more out of the cognitive neuroscience results that we have for the future. And I'll leave you with a couple resources if you're interested in learning more. The first is just a special issue that Rod and I led in Current Opinion Behavioral Sciences a few years ago. There's many different articles from different groups who've been doing precision fMRI work and talking about sort of their perspectives. And the second is a standalone piece, an annual review article that we published just this last year. This is the one where we talk a little bit about how do you even identify networks and compare networks across individuals if they vary so much. So I'd encourage you to check that one out as well. And I'll close by thanking all the folks who I've collaborated with or worked with on these different projects. I've highlighted the names of the people who really led the projects I talked about in more detail today. And I guess I just like to make the point that I think that these precision fMRI projects are really team science projects in general. They often require a lot of time and detailed expertise on various different levels, from the organizational, how do you get people to come in 10, 20 times, to the very practical, how do we align brains as best as we can in order to really understand individual differences and individual brain function. So we need lots of different folks to come together on these projects. And I am happy to continue taking questions. I know some of you already asked some, but hopefully you'll have some more.
We have some questions in the room, I think. Somebody raised their hand. Yeah.
Hi, great talk. So I have a couple of questions. So I firstly wanted to know what your thoughts are on the HAPS-B hyperalignment algorithms, and to what extent the alignments between subjects you can get using that kind of transformation transfers to what you see in all of these experiments and in practice. So yeah, I wanted to know that. And the second question that I have is to what extent you think, or I mean, maybe you already have data from naturalistic long movies and things like that, but I was working with the Algonauts data set, which is like this, basically they have these Hollywood movies and people are in the scanner watching them. And I was a bit surprised to find that for the kind of things I was looking at, the subjects were more aligned than I had thought. So I wonder if you have experiments that are relatively short controlled, if the alignment is less than when you have sort of uncontrolled long stimuli, and maybe as people are just watching for long periods of time. And also I think movies are designed in a very certain way to make people focus on certain things. They are very intentional. They use very intentional choices about audio and visual to make people focus on certain things. So maybe it's that, but yeah, I wonder what you think about long uncontrolled versus short controlled stimuli impacting subjects' alignments. Thank you.
Yeah, those are great questions. I feel like I'm going to try to pull them apart, but feel free to also remind me at the end if I'm not getting to all the pieces. So for the hyperalignment piece, I think this is a great question. It's actually one that tends to come up at NeuroHackademy and in our lab multiple times, and I just have not had someone sit down and do the project yet. But I'd be really excited to try it out and see how much it would account for some of these individual differences. I've talked with Jim, and actually there was like a special session at Dartmouth last year that he organized where a bunch of us who do different types of individual functional alignment came together to talk about these issues. And Fei Long Ma, one of the former postdocs in his lab, he's now got his own lab at USC, I think, has done some work actually directly using hyperalignment methods to look at how much they can improve brain behavior prediction. And what he found in his data set is you could kind of hypothesize multiple ways, and this is something we think about a lot too. Like if you hyperline first, maybe you've taken out the individual differences that you think will then predict the behavioral variables. But on the other hand, if you hyperline, maybe you take out some of the noise in your signal and you improve the brain behavior prediction. And maybe it matters exactly what you're looking at. And so what he ended up finding in his paper, which I think is a cerebral cortex paper from 2021-ish, is that it improved the prediction of, I think it was general executive function variables in the Human Connectome Project to have hyperaligned the brains beforehand, and it produced a pretty good improvement. So I think it suggests that you do want to address some of these individual differences. I do think there's in practice that when you look at hyperalignment compared to, say, individual network mapping techniques, that they're operating on a very different scale. So hyperalignment is really lining up things that are fairly close in proximity within a couple millimeters, whereas when we're looking at functional networks, individual functional network mapping, we're often talking 10 millimeters or more sort of differences even across these different regions and patches. And so I think there might be value to like bringing the two together to kind of align on both the coarse and the fine spatial scale. I know that's something that they have been thinking about as well, but I don't know that we have or anyone has done exactly that yet, but I think it would be a really good approach to go after. In terms of the question of like movies and what we gain from movies and whether different types of states maybe constrain the brain and make people more similar to one another. We have tended to not include movie data in most of our data sets, partly because of time, I guess, and that we didn't have specific questions about the movies. Whether movies improve people sometimes use movies a lot in data sets because they think it'll be nicer for participants to watch and will also reduce motion and sort of accessibility. And I think is true, although interestingly seems to differ a fair amount across people and ages. So Deanna Green's done a more systematic study on this, showing that some age groups really are benefited by using movies and others less. The movie data will change the underlying functional connectivity a bit. I've done a fair amount of work I didn't show today showing that functional connectivity actually overall tends to be pretty stable across different tasks. But with movie data, you don't have the ability to regress out the task as easily. And so I think you get more of these sort of evoked effects that are driving some of the functional connectivity differences in the movie, or at least it's harder to account for those, which is why we haven't done it quite as commonly. But certainly for intersubject correlation effects, and if the question is like, I can't get my subjects to sit still unless I have them watch a movie, I think there is a lot of potential for those approaches. And we actually collected a movie data set in that PAN project, the frontal one that I talked about, the prefrontal cortex project. We haven't done anything with it, but it's available for anyone who might be interested. And we probably will do something with it. Partly it was exactly for the hyperalignment questions you're asking that we sort of envisioned ourselves doing some analyses in that direction. So I think, I guess that's to say, I think there's a lot of potential. I think tasks in general don't change functional connectivity topography too much, but there are like sort of detailed aspects of that, especially with movies, that are worth considering of like, how do you deal with the evoked effects of the tasks?
So I see one of you have your hand raised. Hi, go right ahead.
Okay, thank you. It was great talk. Hi. Um, I see, it's great work, and I'm pushing my lab's research also to the precision mapping direction. But kind of, I, when I kind of see the results and your kind of perspective approach, I kind of feel that your approach is quite different from the training that I got from my PhD for my training. So from my training, for example, we are so into this high dimensional representation view. Related to the hyperalignment question, but like Hopfieldian view, like all these on in the high dimensional space, um, even though the low dimensional kind of representation activation patterns could be different, but in the high dimensional space, it's there is a information. For example, the representational drift is a good example, I think. Like in the low dimensional neuron level, yeah, the exact location could be different, but the high dimensional kind of the geometry is the same, right? And so I kind of really curious to hear your thoughts on how kind of you integrate these two kind of approaches. Like, I mean, I would say, I wonder if I could put this approach as a like geographical kind of brain geographical approach, like kind of comparing the shapes and borders, analyzing the borders, and like comparing building a spatial model, right? Versus this high dimensional representational model. I mean, these two seem a little bit different, but I can see they could be really complementary. But I want to hear your thoughts on, yeah, this. Sorry
Yeah,
for.
I think it's no, I think it's a great question. I think it's hard to do both at once. In some of the earlier work that I did, I started in sort of more visual attention projects where I think both we already did the precision sort of style work there with lots of data from individuals, but it was more focused on representations and thinking about representations and how they change and how they differ. And I think representational drift, like in the hippocampus, is really fascinating, right? Like we still know where we are, but like the individual neurons are changing and how they're coding the information. And I think it's like it may be an interesting case where it sort of links to these ideas that we've started developing. Like we see these network organizations that are very stable over time, and I think those must be at the kind of higher level where again there's these like homeostatic sort of plasticity mechanisms that as a whole I don't think they're going to change that much in terms of organization. But I think it would be really interesting to pair that with analyses that look in more detail at the representations within areas. Of course, it'll still be at like a multi-voxel level, but to see how and to what extent those align with one another. And there's a couple ways that we're thinking about this in the lab, that I've been thinking about this at least. And partly thank you for sending Young in my way because I think she may be involved in some of these projects where one of the things I was interested in is taking a little bit more of a representational or deep look at that pan data set, especially at those border regions to see, beyond just they're activated more in some of these tasks, is there any sort of additional representational information that we can tease apart in certain borders compared to others and sort of why certain activations are happening in certain places. And the other place that we've started thinking about this a little bit in the lab is actually through a collaboration with Adam Steele, who's an assistant professor here. He's done a lot of work on sort of visual to memory transitions within one of the default mode networks and thinking a lot about both the topography of the network, so why are they lined up with one another, but then how does that also relate to, for example, population receptive fields within the networks and how they're interconnected with one another. And so I think his work is a really nice example of trying to bridge those different scales.
Thank you so much.
Right, so I think unfortunately we're out of time for this session. So let's thank Caterina again for this excellent talk.
It's my pleasure. So feel free to also send more questions my way if you have them on Slack or via email.
Exactly what I was going to say. Thank you. Okay, as a reminder.